Good morning, everybody. Welcome from my office again. Happy Friday. Woohoo! Cha, 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 cha. Super excited. Okay. Ba-ba-dum, let's get this up. And two things before I forget. I can spell big. Come on. Tell it's Friday. Big thing number one is the exam one is moved to next Friday. Which is, what day is that? 17th. So the schedule's already been updated and uploaded and adjusted and stuff. So yeah, just to kind of reiterate that. Second thing, I don't know if it was this class or the other section, it's kind of hard to remember. I think thing number two, who asked about transcripts of the audio walkthroughs? So I don't know, again, I don't know if there was anyone in this class. I don't know if it was, if they're actually in attendance or not. But if it was someone on here, can you let me know? Because I need to talk with DSS about possibility of getting that done. So yeah, that's pretty much it. Just figured I'd ask those before I forget. Does anybody have any questions about anything that we've covered up to and including last class before we move on to cell division? Let's give you a few moments. I assume people are there. Yep, there are people there. Give me time to drink some coffee too. Okay. All right, so let's begin cell division. Yeah, I get to read my emails. Here we go. All right, pretty cell, pretty image rather. Okay, so I think essentially there's going to be a few additions to our language, right? How we talk about cell division and DNA and stuff like that. So I'm going to go through those first, just so that you have all of that kind of ready to go, essentially. Right, and so, yeah. Cell division is really what we're going to be focusing on for the next 3 lectures, right? And that's going to take us right up to and including exam 1, right? So this is the last topic of this section for genetics, and then we'll move on to the second section, the second quarter. So cell division is the growth, DNA replication, DNA division, or partitioning. No, not that. And cytoplasm partitioning. Oh, sorry, partitioning is actually a hard word to type. Cell. And depending on whether it's meiosis or mitosis, so mitosis equals, and I'll get into why this is a kind of a tricky word in a sense. Generation of 2 identical diploid daughter cells. Meiosis is generation of 4 non-identical haploid daughter cells through 2 rounds of cell division. So one of the tricky things about cell division and mitosis is that kind of mitosis refers to both the cell division bit above, right? Generation of 2 diploid identical daughter cells, and the DNA division part of cell division, of that cell division. And that's also called M-phase as well. Right, so I'll be very, very clear what it is that I'm talking about when I talk about this, both in class and also in the exam. But bear in mind that that word kind of has 2 related meanings, but means slightly different things depending on the context. Okay, so some other words that are going to be really important. Homologous chromosomes. These are almost identical chromosomes, one of which you get from your mother, and one you get from your father. So they have the same genes, well, they have the same structure, size, genes, etc., but may differ in the alleles they have for those genes. Right, so essentially they're very, very, very similar. Like very, very similar. There aren't really that many differences between them. Obviously, it depends on what the genetic ancestry of your parents are, you know. So mine is fairly similar. They're both from London, in it. But my daughters are likely be fairly different because I'm from the UK, you know, so Northern European ancestry, Scottish and German, and maybe, I hate to admit it, some French. Whereas my wife's from South America, so they're going to have inherently different alleles for those genes. Now, there's another thing which is often confuses students, and this is sister chromatids, right? And so these are identical copies of a chromosome due to DNA replication. And they only exist until they're pulled apart in anaphase. This is where students get kind of tied up in knots between homologous chromosomes and sister chromatids. So essentially, in a normal somatic non-dividing cell, you'll have 2 chromosome 1s, essentially, one from your mother, one from your father, right? During cell division of that cell, each of those are going to get copied. So now you have sister chromatids, so 2 copies of chromosome 1 from your father, 2 copies of chromosome 1 from your mother. Those only stay in that state. It's no extra information. That's like photocopying a book. you don't have any more information or new words. It's exact copy, right? And that only stays in that configuration until anaphase of the M phase, where they get pulled apart. Obviously can't do that because I like my fingers being on the same hand, right? So really keep that in mind. It's a constant source of confusion amongst students. Not everybody, but some struggle to kind of get their head around that difference. And it's a very important one too, particularly in meiosis, right? Where we're going to be separating homologous chromosomes first and then sister chromatids second. What else? Let's see if I get auto-corrected here. Yes, okay. Another important thing to talk about is cohesin. This is a protein that sticks the sister chromatids together until anaphase, right? And so that's what allows them to be pulled to the center of the cell by the spindle fibers, and then it's kind of destroyed to promote anaphase, and that's destroyed by a protein called separase. And when that's activated, destroys cohesin. All right, so there are other words that are going to be important. We'll bring those up as we come to it. These ones are especially important because they're very much central to how all of this works. Okie smoky. Okay, so before I get into any of that, I really want to talk about the cell cycle, because this is a super important part of both mitosis and meiosis. Okay, so here there's two main points that I want to get across here. I'm going to spend a bit of time on this because there's a lot of information here, and it's a lot of key parts of cell division are represented here. So this is also for eukaryotes. So, animals, plants, things like that. Prokaryotes have a different cell cycle because they reproduce by binary fission, right? So, and also they don't have a lot of DNA that needs to be divvied up, right? So it's a little bit simpler for bacteria, but for eukaryotes, it's a pretty tightly controlled dance to get the outcome that you want. Okay, so the first big deal is most of cell division is preparing for the division of DNA. Right. So the division of DNA is this bit here, M phase, or really mitosis, right? Part of M phase. And you can see that this is actually a fairly reasonable kind of pie chart in a sense of cell division. The vast majority of the time in cell division, which for mice, I think it's about 24 hours to go from entry into cell division to 2 daughter cells. Of that, a couple of hours are spent in M phase. Right, so 80 to 90%, depending on the organism, of the time that it takes to divide a cell is spent preparing for that last step. And so that other bit is called interphase, right? And it's both cell growth and preparation for everything that needs to be done. Just calling it cell growth is a little misleading because there's an awful lot of other stuff going on there, including DNA replication, which obviously you need to be able to produce 2 identical daughter cells. You also need it in meiosis too, but we'll get to that later. Of interphase, ooh, don't want to lose that. That's about exam 1. Of interphase, it's split into 3 different phases. right? There's gap 1, G1 is called gap, or these are called gap phases because it looks like nothing's happening using the microscope, but obviously stuff is. Gap 1 phase is all about preparing for DNA division. A little bit of growth, but mostly about DNA division. Because S phase is where all of the chromosomes in that cell get replicated. Right? So you end up with twice the amount of DNA in that cell, not twice the amount of information, twice the amount of actual, if you were to weigh it, massive DNA. Then once that finishes up, gap 2 is all about preparing for M phase or mitosis. So all the proteins, and there are many that are needed for that to go successfully, are made in G2. Right, so that's kind of the big bulk of cell division is interphase. Right, so you can kind of see stuff happen. Cell gets bigger. You know, if you're staining for DNA, you'll see more of it. But the action, that only happens in the M phase part. Okay, so that's kind of one big point about the cell cycle. The other one, and I actually tend to spend a fair amount of time on this because it has a lot of real serious implications for biology in general and also human health, is that this cycle is exquisitely controlled. It is, if you think about it, right, if you think about it in the context of, you know, you as an organism, you're the result of thousands of rounds of cell division that happen exactly where they're supposed to, one hopes, at the right time, the right amount, and they stop when they're supposed to, too. Right. And so cell division is extremely tightly controlled. And that also means that when that control slips or lapses, problems happen. So just as a kind of question to the class, what do you think happens if these cell cycle checkpoints stop working? What's the most, you know, likely disease that you can think of? And you may have had a relative or friend of the family or something. Yeah, exactly. Cancer. Right. So cancer is essentially the uncontrolled cell division of a particular tissue. Right. And so the checks and balances that stop that happen, that lock it down, because a big chunk of your body is not dividing. Right. And it shouldn't. Right. I've already got a set of lungs. Why do I need more lung tissue? Right. And also the cues, the signals that tell specific cells, you're going to divide here and you're going to be this, right? So your, you know, lung cells, you're going to divide, you're going to become an alveoli, like a kind of a planar squamous sort of epithelial cell. Those signals are not present in an adult, not in the right way or the right context. And so that's why when, you know, you talk about cancer, we're not talking about, you know, growing another arm or, you know, growing another kidney. We're just talking about uncontrolled, unspecified cell division, right? Because all of that context, which, you know, generated you over, you know, a period of months and years, doesn't exist anymore as an adult, right? So these cell cycle checkpoints are extraordinarily important. Okay, so there are 3 of them. We have the G1, oops, to S checkpoint. And that is all about Well, a bunch of different stuff. And you can pretty much divide up into, pardon me, one, does the cell have enough nutrients, energy to make it all the way through cell division? Because if you don't make it, the cell dies, right? So if the cell pauses at any point here, other than the spindle assembly checkpoint, which is kind of different in some ways, then typically the cell will be told to kill itself. And that's apoptosis. A process called apoptosis or programmed cell death. And that's actually a process that's really important in development. So this is a kind of a fun fact that you can, tell your family or friends or whatever. But when you're born, wee baby, you had more neurons, about 60% more than you do now as an adult. Isn't that funny? So in some senses, you're actually dumber than when you were born. Obviously, that's an oversimplification. And so those excess neurons are killed by apoptosis. It's called neuronal pruning. Right? You're just kind of like chopping off the bits that you don't want anymore. And so that actually kills a good 20 to 40% of your neurons. Basically, because you're not using them. So there you go. The whole, yeah, no, yeah. Getting dumber every day. Actually, that stops by, I think it's about 5 to 6 years old. I'm not actually 100% positive about that, Caitlin, to be honest. Hang on. Go away. Because I'm pretty well up on many aspects of development, but my kind of interest in development pretty much stops at birth. Kind of childhood development isn't something. It's kind of like an extension of what's already set in place. I'm actually more interested in how do you get to that point. Well, the funny thing is, Bianca, it's not about being smart. It's actually about being efficient. And so essentially what it is that you have way more neurons than you need. And the ones that form connections with other neurons, basically kind of reinforce those connections and form that neural network, right? That's actually how you start being able to, think and have coherent thoughts and stuff, not just like, oh. Hang on a second, Isaiah. So the ones that aren't needed, essentially the excess, are kind of being, just killed off because you don't need them. They're not doing any good. Brains, I don't know if brains are getting smaller, Isaiah. Certainly your brain doesn't physically get smaller. Obviously it gets bigger because, if you compare the size of my head with, a baby, if the brain stayed the same size, it would just like rattling around in there like a tennis ball. So brains do kind of grow in size with you in terms of development. That's why partly one reason why the plates don't fuse in babies' heads for a little while, which is kind of neat. Also, so they can, squeeze their way out, so to speak. But yeah, this process, apoptosis, is really important in development, but it's also very important in preventing uncontrolled cell division and also division of cells that shouldn't be divided, basically. So, and also another point is, say neurons are actually a really great example of cells that are what are called post-mitotic. So once a neuron is born, which is kind of how we refer to it, it will never divide again. You know, a cell that is born that will become a neuron, that's it. Other cells, glia, I think, support cells, I'm not sure if those divide again or not, but neurons at least, once they're specified as a neuron, that's it. They'll never divide again, right? Because these checkpoints just clamp down and completely lock out all possibility of cell division, at least if they're working. And so the other thing that the G1 to S checkpoint checks for is DNA damage. Because if the DNA of that cell is damaged and it manages to make its way through DNA replication, that damage will get copied. And if it makes it through the G2 to S, G2 to M checkpoint, the two daughter cells will have that damaged DNA. So basically making sure that only fit healthy cells, in essence, make it through into cell division is a really important part of kind of quality control, essentially. Right. So the G2 to M checkpoint, right, which is after DNA replication, because that happens in the S phase. This is all about checking for DNA damage again, and completion of DNA replication. So essentially making sure that all of the DNA has been copied and none of it is damaged, right? There are no broken chromosomes, there are no regions where there are no bases, right? Single-stranded, stuff like that. Super, super important. And if there is damage, sometimes it can be repaired. Now, here's another kind of real life sort of context type question. Does anybody know what these two genes are involved? Associated with? This may be something that particularly the ladies amongst you will have heard of. Damn, that was fast. I didn't even have a chance to drink my coffee. Yes, Savannah and Infinity, breast cancer. Right, so BRCA1 and 2 are involved in DNA repair. So if you have if you're homozygous recessive for both of those, essentially if you have DNA damage, and I don't know why this is tied with breast cancer specifically. I don't know those details. Let me. I don't know. Let's have a look. Jack. Oh, that's not a big help. BRCA1 DNA repair associated. That doesn't really help. Sorry, I'm just kind of curious. Yeah, breast cancer 1 and breast cancer 2. I guess it's in the name, right? So if you have, and there are genetic tests that you can have done for this, particularly if you have a familial history of breast cancer. But if you have non-functioning copies of both of those, then your risk for breast cancer, right? And this isn't a certainty, this is just probability if you look at a big enough population. is about 80 to 90%. And I think the most famous example that would be Angelina Jolie, who's an actress, UN ambassador and stuff. And she had a double mastectomy, right? Because she wanted to head off the possibility of breast cancer. She also had a familial history of it too. So, you know, these checkpoints are extraordinarily important, right? And mutations in checkpoint genes or genes involved in DNA repair are what one would call are part of a group of genes called oncogenes. So these are genes that, when mutated, and this doesn't mean that they don't work, they might be mutated in a way that makes them work when they shouldn't, right? Cell signaling genes, for example. Basically, when they're changed, their function changes, they promote cancer. Now, that might be mutations in tumor suppressor genes, right, of which BRCA1 and 2 are part of. So they're involved in preventing tumors as part of their normal function. Or it could be things like growth signaling genes, which are normally the signals that tell cells to divide. Actually, my research, a large chunk of it is about a gene which, when activated, promotes lung cancer. So, for example, I'm not a cancer person at all. I work on worms; they don't get cancer because they, for various reasons. But anyway, so that's actually part of my research. And these two checkpoints are controlled by a whole bunch of other stuff. I was actually reading up on various checkpoints because this is actually something I kind of know about, but you know, not as well as I probably should. Look at that. You can't even see all of it on a page. So this is essentially the process by which these two complexes, so there's cyclin-dependent kinase 2, which I've worked on, cyclin E, and so on. Cyclin because it's part of the cycle, the cell cycle, right? And it cycles, it oscillates in terms of its concentration. These are what altogether control that first checkpoint, right? So don't worry about this. I'm kind of putting it up there to like shock and awe, right, tactics. I'm not going to test you on any of this, right? But just to show you that this is a seriously complex process, right? And it's very, very finely controlled because it's super important that it works properly. Yeah, I'll take that off. There you go. I'll read that later. So the final one is the M-phase or spindle assembly checkpoint. And this checks for the alignment of all of the chromosomes on the metaphase plate, which we'll get to what that is in a little bit. And so it's just, and I did actually have a little bit of fun playing around with the drawing thing on Zoom, but it's a bit of a pain in the ass. So I'll actually just use the whiteboard. Ooh, stretch my legs. So the M-phase checkpoint, its role is to make sure that essentially all of the two daughter cells get an equal number of chromosomes, which, as you can imagine, is very, very important. And this checkpoint also applies in meiosis too, and we'll get to something cool in there. So essentially, you know, if that's a cell, that's painted in dividing cell, and we have our centrosomes here. So this is a centrosome, right? Or an mTOC, and I'll write out what that is in a little bit. Essentially, you have your chromosomes look like so. These are individual chromosomes. These are sister chromatids, right? Because you have two copies of the same thing. And these are going to be attached to the centrosomes by spindle fibers, right? Again, don't worry about the details of this because we'll be covering that in a little bit. And so the role of the M phase checkpoint, these always think of like little marshmallows whenever I draw centrosomes, is to make sure that those chromosomes or chromosome pairs in meiosis 1 are aligned on this kind of imaginary line called the metaphase plate. Right. Once that condition is met, and only when that condition is met, are these allowed to be separated and get pulled into opposite sides of the cell. Now you can imagine, so this is the check is good to go. Now this is a totally random process, right? This is actually based on mechanics more than it is, biology or biochemistry even. So you can imagine what would happen if instead, say we have this situation, right? So we have a chromosome that is, maybe it has only a spindle fiber attached to one side, not the other. Maybe it has two on one side, one on the other. Maybe it just hasn't made it to the center yet, right? What would happen if this cell was allowed to go through M phase and finish dividing? What would the result be in terms of the two daughter cells? What's the consequence of that? If instead of this being paused by the M phase checkpoint, the M phase checkpoint goes, yeah, looks good to me, and gives it the okay. What would happen in terms of the two daughter cells that are produced from that? Particularly in terms of the number of chromosomes that they have. Yeah, exactly, Stephanie. One would have two of this one and one of each of those. This one would have three and none of that one, right? So this is, if this were allowed to occur, and this is actually normally, blocked, this would result in something called aneuploidy, where the cell has one too many or one too few chromosomes. Now, let me just type that up. And while I do that, can anybody think of a human condition? It's actually fairly common, about 1 in 1,000 births. That is a result of aneuploidy, the M phase checkpoint not working properly. Yes.
Is it
Go on, Rebecca.
I'm trying to remember what the name of it is. Special needs.
Kind of, you're getting there. It's Down syndrome.
That's what it is.
Yeah, so special needs in general can have many different causes, but one of the common causes of child needs special needs, I guess. I don't know. I haven't really thought of it that way, is Down syndrome. Let me just finish typing that up. Right, so exactly, Stephanie. Down syndrome is the classic example of that. So Down syndrome is it trisomy 21? Let me just double check. Or was it 23? Yes, 21. And so the crazy thing about that is that for all of the women in this class, well, all women in general, girls also, is all of your potential oocytes, you know, what will be all of your oocytes that may become eggs are created while you're still in the uterus, you know, it's still inside your mother. And actually they are also pruned, I think, I can't remember the exact numbers, hundreds of thousands maybe of them get knocked off to give you what you end up with, but they're all paused in the first round of meiosis at this point. So essentially, if you do end up having children, that child is a result of an egg which completes meiosis after being paused at this M phase checkpoint for however many years since your birth until you have that child. Right, so you can see that as you get older, the risk of a cell slipping through that checkpoint gets greater. And actually the risk for that happening in males also increases with age. Down syndrome isn't necessarily just a result of errors in cell division with in women. But yeah, and you know what's even more insane, Kaden, is that Down syndrome is a very, very mild version of aneuploidy. And there are, I actually have to make a list of this and write up on the wall. I think 4 or 5 chromosomes where you can have an incorrect number and still get a viable embryo, a viable fetus. All the rest of them, that embryo will not make it to full term. It will, you know, essentially be a miscarriage, because an extra 1 or 2 of any of the other chromosomes is lethal. It results in, you know, not developing properly. So just to kind of show that, you know, Down syndrome is a very clear and obvious syndrome, right? You just look at an individual and you can tell pretty much instantly if they are Down syndrome or not, because I have certain, you know, facial features and behaviors and stuff which you know, are fairly easy to see on the surface at least. And so, yeah, anyway, that's something that always blows my mind. Okay, so anyway, that's kind of a lot of time spent on this, but it's also, in my mind at least, such a cool part of biology and development and human health that I think it's really worth spending time on. Okay, so now we've got to move on, because otherwise we won't get through all the material for the exam. And I'm just going to skip ahead essentially to here. Yeah, I think it's super neat, Infinity. I really do. It's one of the reasons why I enjoy teaching developmental biology so much, because it's just fascinating. It really is. Okay, so this is what your dividing cell, right? So it's already committed to dividing as a G2. It's duplicated the chromosomes. The centrosomes have duplicated in G2, chromosomes in S phase. Now, just to get you thinking, what needs to happen next? Or what needs to happen just in general to be able to divide that DNA up? Just want you to think about looking at that picture. What do you need to happen to be able to pull, pardon me, those sister chromatids apart? Okay, cool. First thing, centrosomes need to be opposite each other at the poles, right? So you have 2 centrosomes here, they will push themselves apart. They actually do so by using spindle fibers. So they're used to push the centrosomes apart, then attach them to the plasma membrane. My typing is not very good today. And essentially lasso. I'm not sure that's spelled right. Lasso. The chromosomes. Yeah, just before we go there, I just want to show you what microtubulin looks like because it's a pretty cool protein. Get that out of the way. So microtubulin is actually a polymer, right? It's a repeating spiral. Actually looks kind of like a churro, to be honest. Consistent of two different proteins, alpha tubulin and beta tubulin, right? And these can assemble, really nice, pretty confocal microscopy picture there, and shorten, right? So you can make them longer, you can make them smaller, and you can actually kind of apply tension with them. And so you can polymerize them, add in more units at one end, and then you can kind of take those units out and shorten them. That's how we apply tension to the chromosomes to get them to line up in the middle in metaphase. So it's a pretty cool protein. It's also the kind of like the highways of the cell. So for your neurons, which are great, you know, so there's a neuronal cell body, ganglia at the base of my neck, and the axon for that goes all the way down to tip of my finger. And so the proteins that are required down there for, you know, for the neuron to work, like synaptic proteins, are carried down there by molecular motor proteins called kinesin, well, carried down by kinesins, carried back by dyneins. And these actually walk like this. They're really cool. Anyway. Okay, so what else is needed? What has to happen as well? So first, we've got to get those centrosomes out. Exactly. We also need to condense these chromosomes, right? Because this is like a bowl of spaghetti, really hard to kind of pull it apart. So we have to condense the chromosomes down to be able to accurately partition them. How do you get to those chromosomes? We have a nuclear envelope or nuclear membrane. What's got to happen to this? Can it stay there? No, it's got to dissolve, right? So the nuclear membrane has to disappear. And finally, the last thing that needs to happen is you have to have some way of attaching those chromosomes to the centrosomes, right? So those spindle fibers have to form. So the bulk of M phase, right? So again, we've talked about M phase is like a small part of the cell cycle, right? The bulk of that small part is all of what we've just talked about. Getting the cell ready, or the DNA ready, so that it can be pulled apart in anaphase. So most of the time in M phase is spent doing all of those things, right? And aligning the chromosomes on the metaphase plate. And essentially telophase or telophase, which is the kind of the end, is reversing all of those steps, right? So you kind of can think about, you know, M phase as being having two bookends, right? You start here with a picture like that, you end here with a picture that looks pretty much like that, but it only has one centrosome. You kind of go from and then back up again. All right, that's enough for today. Have a look through the rest of it. And also, if you can, have a look through the meiosis PowerPoint. It's way more complicated than mitosis. There's a lot more going on. And so it would really do you good to kind of have a jump on that. So we're going to finish this off on Monday, and then we're going to spend the rest of Monday and Wednesday going through meiosis, because there's a lot of stuff going on there. Awesome. That was a lot of fun. Hope you enjoyed it too. I'm going to get off and go hassle IT, and have a great weekend. I'll see you on Monday. Oh, Jessica, fire away. Do you want to type it up, or you want to wait until people have left? Or if you want, I can send you a kind of like a private room invite. It's up to you. Ah, no, I couldn't hear you. Sorry. I don't know if you're unmuted or not. Yeah, you are unmuted. Try again. No, I can't hear you. Tell you what, I'm going to end this, and then I'll send you an invite for like, oh, I thought I replied to that. Give me just a moment. I'm going to end this just so, you know, file doesn't get too long. I'll send you a invite in just a moment, okay? Won't be a sec.